Hello, and welcome back to Beyond Networks, the Evolution of Living Systems. In last lecture, I went through a very extensive detailed description of my own work with colleagues, of course, on the gap gene system in Drosophila and how we used both, decomposition, genetic decomposition of mechanisms with modeling to reconstruct the causal flow in the gap gene regulatory system. Sort of the gist of that analysis is summed up in this quote from an upcoming paper that I'm writing with philosopher James DeFrisco. And we're writing dynamical models properly formulated and validated, with a genetic interaction matrix, it's fit to data, it's validated with experiments, provide the tools to probe cause and effect relationships in complex biological systems, allowing to go beyond the limitations of genetic reductionism to gain an integrative understanding of the causal processes underlying complex phenotypes. So not only a parts list, but an understanding, a processual understanding of what the system does and also what the system can do to a certain degree. So in this spirit, we're going to link this sort of investigation that I was introducing last time back to the earlier ideas about causes as difference makers that I've introduced a few lectures back. So here is this picture of classical genetics, which points out that in those specific populations of fruit flies that Morgan's lab was studying, there were different eye colors that you can see in this beautiful drawing from Muller, and the actual and specific difference makers for these eye colors. So the factor that actually varies in these populations and that is specifically responsible for causing the different eye colors, those factors are different genes that Morgan and colleagues obviously could identify and founded classical genetics. So classical genetics is based on this sort of very, very powerful interventionist counterfactual approach, which I will call the difference-making gene paradigm. So this is the actual factor that's specifically changing the eye color if you intervene on it. If you mutate the gene, the eye color will change. And if the alleles of the gene vary across a population, you will have different eye colors. Okay, so how does this work for more complex situations? This is a very simple trait. Most complex disease traits, behavioral traits, and also most developmental traits are not as simple as that. So let's have a look at those. And let's sort of connect back to Waddington's metaphor of the epigenetic landscape, especially this part of the diagram that shows the underbelly of the landscape. And Waddington's powerful metaphor of this network of guy ropes that connects the pegs of the genes to the topography of the landscape. So you want to understand what is the effect of removing or changing one of these pegs on the topography, the shape of the landscape through this complicated network. It's a very difficult task. Or in a bit more modern terms, you have a depiction of the genotype-phenotype map here with gene regulatory networks underneath, these sort of molecular regulatory processes and all that, phenotypes on top, and then this space of dynamic regimes in the middle that mediate this complicated effect. So that corresponds, that part in the middle corresponds to this network of guy ropes in Waddington's picture. And this is what Waddington called the epigenotype, the epigenotype being between the genotype and the phenotype. So how can we apply this idea of counterfactual causation as genes and other factors as difference makers to this more complicated picture? That is the question we're going to address here in this lecture. So first we need to be very explicit about how complicated this task is. So most developmental processes not only involve gene regulatory networks. This is often overlooked nowadays. So this is only one level of complexity here, okay? So you have underneath the gene regulatory networks, you have metabolic processes, physiological processes that are happening at this, below the cellular level. Then you have the cellular and the tissue level, at which you have cell-cell signaling, local interactions between cell-cell movements, cell shape changes, et cetera, that can feed back on gene regulation. And even higher up, you have the organ. Here is one of my favorite organs. This is a human liver, and you have different tissues that integrate into this functional whole of a detoxifying sort of station for your body, which is a great thing. And that, again, is part of a larger sort of integrated organismic system, okay? And these levels are all sort of feeding back on each other in a very mysterious way. We're going to spend the whole next module looking at these different levels and how they interact in more detail. But we're going to start looking at this sort of multi-level nature of regulatory processes right now. And of course, this is all embedded in an environment. Environmental triggers can influence gene expression as well, or physiological states, as in this example, you drink the beer and you get a little dizzy and happy. And so you have this very complicated multi-level system that's heavily driven by feedback. That's very important. So this feedback regulation and multi-level interactions, I would say, are sort of the defining characteristic of these sort of complex regulatory processes, for example, developmental processes that we are most interested in this lecture. Okay, so how does genetic causation fit in here? And what's happening? What is the context of genetic causation in this broader picture? That is the question. Okay, so let's start with sort of a few initial observations, such as, as systems biology goes beyond the limitations of genetic reductionism, we'll find two basic challenges. So this lecture will have two parts. The first will be dedicated to how do we reach an understanding of causality compatible with the heavily feedback-driven dynamics of regulatory processes. So when the flow of causation becomes circular, becomes a bit of a headache, deconstruct how what is causing what, and we need to pay attention to this. The other factor here, which is different, is how do we provide an understanding of gene action in systems with a multi-level structure? So we take these apart. So let's look at feedback regulation first. And we can do this by using our old friend, the toggle switch model, again, to illustrate issues. And the first thing we're going to do is remember, there are two genes here. They repress each other, double negative. It's a positive feedback loop. So what you get is either X on or Y on, but not both of them at the same time. You can have some autoregulatory interactions here of the genes on themselves. There is some constitutive activation, and of course, in a dynamical system, the factors also the proteins need to degrade. So if you tune this autoregulation, remember this very simple network will enter into different dynamical regimes. So it can be either bistable, or when you increase autoregulation, you get a bifurcation and you get a new attractor here in the middle where both factors can coexist. So you get a tri-stable system. Now, the main point that you need to take home here is that most of these regulatory networks, even a very, very simple one like toggle switch, are multi-stable. So this happens in most nonlinear systems, okay? And so if you have such a multi-stable network, the effect of a genetic intervention depends on the phase space topology. If you push the system around, if you change the parameters where you are, if you change this system or this system, you will get different effects, okay? It depends where you are right now. So it depends on the genetic background. We'll get back to that when we talk about epistasis and evolution. This is quite straightforward. But what this means for sort of the gene as difference makers account is that the causal roles of individual genes, they become unstable at this level because they become sensitive towards the state of the other, the state of the whole network, basically. Okay, so as explanations, they are no longer stable as explanations for the behavior of the whole system. Okay, so the switch-like behavior, which only occurs if you have two genes, of course, in the toggle switch with two genes in this case. Okay, so if you want to explain the behavior of the whole system, you have a problem. One gene alone will not explain that behavior. It will depend on all the other factors and interactions in the network. So genetic explanation is highly sensitive to context. So we formally established that right now. So if you put that in a more realistic example, the example of the gap gene system that we looked at last time, if you have a factor, let's say cruple, that is involved, look, in all 3 different AC/DC circuits, if you mutate cruple, the effects that it will have on the circuit that is in a switching-like dynamical regime will be very different from the effect it'll have on the circuit that is in the oscillating regime here. So one mutation will have very different effects in different contexts, in the same animal, in the same developmental process, because the different parts of the network are in different dynamical regimes. So, you know, that's what I just said. So whether you're in a switch-like or oscillating behavior, the effect is going to be different. Another complication that I haven't brought up yet, but biological regulatory systems are very robust, right? And so they achieve this robustness through crosstalk between regulatory processes. They can do this in 2 different ways. It's a beautiful paper by Andreas Wagner in 2005. This figure is from a later review by Marianne Felix and Andreas Wagner in 2008. And it shows 2 different ways in which regulatory networks in biology can achieve robustness. The easy one is actually on the right here. It's just a simple engineering solution. If you make a pathway redundant, it'll be robust against perturbations in any or knockouts of any of these factors, of course, because you always have an alternative pathway that can compensate. But the situation can be more complicated, where it's not directly obvious. It's not just sort of the duplication of the same pathway. It's not just redundancy. And this type of robustness, where you can have rerouting of specific causal influence along different branches of the network, is called distributed robustness. And it involves all the intermediate nodes of the network in here. So both of these cases correspond to sort of a crosstalk situation between regulatory processes. So if you knock one of the factors out, you can have different processes go around it and compensate. And such type of crosstalk can be synergistic. So you can, if you have 2 pathways, they can boost each other's activity. They can be antagonistic. So one of them dampens the other, or they can be completely redundant. So you knock one out, the other, the system doesn't care, okay? And so this obviously gives us a huge headache for interventionist causal investigation of genes as difference makers. The biggest problem here is obviously that interventions on antagonistic redundant pathways often have no effect at all. So if you have no effect, remember, in the interventionist account, you don't have a cause, okay? So genetic explanations are no longer relevant, or the technical term is proportional, as causal explanations for the behavior of the whole system. But it doesn't mean that these redundant genes here in the network, they don't do anything. There is specific causal influence that flows through these different pathways, and you can track them using that method by Griffith and colleagues. By measuring mutual information, you can track exactly how much information flows through the wild type network, and there will be information flowing here. But in terms of the interventionist account, if you knock them out, they have no effect on the system's level behavior. So in a way, they are not relevant to the explanation of systems level behavior. Okay, so these genetic causes in these systems are relevant and specific at the gene expression level, but not at the systems level. Okay, more complications because of the sort of structure of a complex regulatory network. Let's go back to the toggle switch model and look at the bistable regime. So the classical toggle switch regime, where this feedback here, the positive feedback, dominates the system, and you have 2 states, and they implement the classic toggle switch behavior. Now imagine that you perturb this system at the beginning when you're very close here, you have some low concentration of both factors, it's fluctuating around here, you perturb it, it goes smoothly to one or the other. But what if you perturb a system that's already at steady state? it's going to be really hard to get it out of this basin of attraction here because you're very far away with the attractor from the separatrix. So the intervention, the effect of an intervention depends on the history of the system. This is beautifully depicted in this cusp graph in configuration space by René Thom. Basically, at the beginning, if you intervene at the beginning, you're here. You're somewhere in the middle, you push it up, you push it down, and you get a high or low concentration. Here's the state variable. Let's say this is scruple concentration. Here you have parameters that you change. Okay, but if you move into this regime, where the system is sort of bistable, if you have it on a crucial high sort of state, you need to push it all the way here till it falls down, and it's going to be very stable. Okay, so you need to push it back much harder to switch it back and forth. So when you're here at the front of the fold and you have hysteresis and truly bistable behavior, it's much harder to change the system from one attractor to the other than if you have this sort of smooth behavior in the back here. So basically, hysteresis renders the causal specificity of genetic interventions history dependent. It depends on whether you've already arrived at the stable state or not, and you've already settled into a hysteretic situation or not. And causality here becomes unstable, sensitive towards time. And it can no longer be expressed in terms of atemporal counterfactuals. You cannot simply say, if X were the case, Y would happen. You can only say, if X was the case at time so-and-so, or if X would not have been the case at time so-and-so, then Y would not have happened at that time. But the whole formulation becomes much more involved and complicated. And this is very important because the difference, sort of the simple difference-making representations that we use in classical genetics and also in causal modeling. So classical causal graphs are atemporal. They abstract away from time. And so they are no longer adequate to characterize causation in feedback-driven regulatory systems. So systems that are really dominated by feedback dynamics. And you need a dynamical model. So this is just to point home, this is a different way to drive the point home again, that you need a dynamical systems model to understand the influence of different individual causes over time and space in such systems, okay? And so there's no way around a dynamical approach here. Okay, so we've used this sort of, dynamical systems approach I showed you last time. And I showed you how it enables us to deconstruct the causal flow in such system, but it enables us to do something more, okay? And this is very important here. So what it allows you is to do systems level interventions. What do I mean by that? It allows you to change one of the interactions or the nodes in the system, remember, and to sort of analyze the effects of that, either through simulation of perturbations. So you simulate what the experiments that you would do, or, and I introduced that last time when I showed you how we found these dynamical modules, the AC/DC circuits in the first place, we used sensitivity analysis to see how the system would react to these perturbations in general. Okay, so basically this approach yields a proportional, meaning relevant at this level, stable and specific account of causal flow in robust feedback-driven systems. You cannot get this without dynamical systems modeling. The classical genetic approach is no longer working because the gene action, the action of individual genes becomes unstable, irrelevant because it's no longer proportional, and also the specificity of what's happening depends, for example, on time or on the context of the system. So this is instability, sensitivity to context and time. Okay, this is very important. So our ability to tackle complex systems with classical genetic interventionist methods, perturbation methods, completely breaks down at this level. And even worse, it is no longer adequate or proportional to explain the behavior of the whole system at the level of single genes. We'll come back to that when we talk about network drift. This will become very important. But basically, single genes are not the right level. Never mind the molecular details underneath. You need to explain network behavior, system-wide behavior at the system level, at the network level. Let's get back to this sort of hierarchical, multi-level system now. The gap gene system was very simple, and it only involves gene regulation. So it doesn't have any of the additional complexities here. So we've seen that introducing feedback regulation complicates the picture already. But what happens if we introduce feedback between multiple levels? So let's point out a few things. First of all, the classical approach, again, the genus difference-making, difference makers paradigm collapses all this complexity away into two abstract levels and just maps correlations between genetic variation and phenotypic variation. So you have a mutation, it moves around your organism, your position in genotype space, and that will have an according correlated output at the phenotypic space. But you completely black box everything that's happening in between, okay? And so this is very simplified, because in reality, you have multiple levels of interactions here. And this is very intricate causally, because, so what are these levels in the first place? Why am I talking about levels of organization here? So levels of organization are a very, very complicated and controversial topic in the philosophy of biology. And they're very widespread. We all use levels all the time. Levels of explanation, levels of organization, all kinds of levels. So here we're talking about levels of organization like the genetic level, the tissue level, etc. So how do they differ from each other? The main way they differ from each other is that causal interactions segregate them. So basically there are more causal interactions between entities at one level. For example, molecules primarily interact with molecules, cells with other cells, organisms with other organisms. And so while, you know, you have some much lower level of much lower occurrence, much lower frequency, let me not use the word level here, for interactions between levels. They also occur, for example, entities at lower levels interact with higher levels. Mutations can have severe effects at the cellular, tissue, organ, and organismic level. Death occurs, for example, a very sort of system-wide effect of a molecular mutation event. And higher levels can interact with entities as lower levels. This is also not controversial, because we can have inductive signals that come from other tissues. We can have environmental triggers that come from outside the systems. They come from a different level and alter gene expression at the genetic level. So these come from higher levels, tissue, or even the environment, and alter lower levels. So you have substantial interlevel hierarchical feedback, despite the fact that most interactions happen within level. So how can we, distinguish, how can we find sort of a criteria to distinguish these two types of interactions, within level and between level? So let's look at the within level interactions first. Same level interactions take place between entities at similar spatial, temporal, and force scales. Okay, so they are similar to each other. And so they are very frequent, these events, relative to the time scale you're looking at. So imagine, how do you distinguish metabolic events from genetic regulation? It's mainly a time scale event, right? So the metabolic sort of reactions are happening very fast and are supposed to settle into steady state. And then the genetic regulation is happening at a longer time scale basically. And these interactions often involve direct transfers of matter and or energy. It's quite obvious with the metabolism, for example, but also cell-cell interaction, cell-cell signaling exchanges chemical signals. Okay, so then you have intra-level interactions, which are very different. So they are more indirect in a way that I will explain to you in a minute, and infrequent, or even constant relative to the time scale of a level. So they happen very slowly. And now, and this is very important here, they are often mediated by constraints, which are somewhat unusual causal interactions, because, so what is a constraint? A constraint, that's a very important concept for the next module. So pay attention here. A constraint is a material structure that acts on some dynamic process by reducing its degrees of freedom, but without itself being altered at the spatial or temporal scale of the process. So let me give you an example, that's easiest. So a material structure It acts on a process. It reduces its degrees of freedom. It's not altered itself at the scale of the process. Enzymes, of course. So here you have a map, a metabolic map of a cell, and it indicates the different reactions that occur. But what's important here is that all of these reactions can occur naturally. Okay, you don't need enzymes to catalyze them. Enzymes only change the rates at which those reactions occur. But this is essential. So you can look at this graph and say, okay, you could get a living thing out of non-living matter because all the reactions could in principle occur, but the living organism only emerges from this network if the rates are tuned by the sum total of the enzymes in a cell. And these enzymes, of course, are not affected themselves by the reactions. So they are replenished, they degrade at a much lower time scale, slower time scale than the metabolic reactions themselves. So they're not altered. They are a material structure, of course, the enzyme, the whole totality of all enzymes in a cell. And then they are not themselves altered in the process, and they are absolutely essential for channeling the system through certain preferred channels. Okay, so there are many more reactions that could occur, but by catalyzing these particular reactions, this particular network emerges from the set of all possible chemical reactions that the metabolites could undergo if there were no enzymes. And that distinguishes a living metabolic system from a non-living dead mixture of chemicals. So classical example of a constraint. And we are also thinking of enzymes being produced at the genetic level, so they are supposed to be instantiated at a higher level than the metabolic reactions that happen underneath the set of enzymes of a cell. So constraints usually occur at a higher level, I just said that, than the metabolic reactions, the entities they constrain. And if you look at the whole thing from a dynamical perspective, lower levels determine the initial conditions and the possible states of a biological process. So you have a certain set of metabolites, they're given by the food, whatever the current metabolic state of the cell, and also the possible cells. You have the network of all possible reactions is given by the metabolites alone. The enzymes don't change that map of possibilities at all. So the system has different possible dynamical regimes it can enter, but the constraints sort of provide specific boundary conditions to the system that determine the context in which a process occurs. And so they reduce they change the probability. They don't alter the range of possible behaviors of the system, but of the lower level processes, but they change the probabilities of specific dynamical regimes to occur in the system. Classical example, the enzymes again, right? That's what they do. They don't determine what kind of, they can catalyze an impossible reaction, but they can speed up a reaction that is already possible without them. So, and this is very confusing to certain philosophers, constraints are a form of downward causation. So a system, a whole is constraining its parts and is causing them to behave in ways they would not behave if they were not part of that system. And we'll spend the entire next module worrying about this phenomenon, more or less. Okay, so constraints, their relationship to dynamic process. There is no miracle here. Constraints are just another kind of difference making cause. Okay, so they fit perfectly well in a counterfactual account, just not that specific actual genes as difference makers, actual specific difference makers account. So constraints have a specific relationship to a dynamic process that can be explained in terms of counterfactual dependence. So take this, if enzyme E had not been present in such and such a concentration, reaction R would not have proceeded at such and such a rate. So it's a bit special because it's more specific. It's a quantitative counterfactual, right? So you have specific rates, sort of. It's a continuous variable that can change and have very specific, very fine-grained influence on the process it controls. But they're different in another way also from the counterfactuals we encountered for genes, and that is they relate constraints. The counterfactuals that we use here relate the constraints to inherently dynamic entities, so processes. Okay, what is being constrained is always a process or an activity. And what's interesting is that these processes or activities, they would occur if the constraint wasn't there. So there are productive causes that are sort of the chemical and physical processes that are occurring. And then there are constraining causes. They're all difference-making causes, but they're different in this way. So constraints are different from those productive causes underneath that generate these processes in the first place. Because these processes would have their own causal trajectories in the absence of the constraints. And this is what's called the default behavior of the underlying process. So what a constraint does is it biases the behavior of the process away from its default behavior. For example, the enzyme channels the metabolism in different directions than it would not happen if it was outside the organism, because it would not go in that direction, right? So there's no magic about downward causation and constraints are just different making causes, but they're of a different kind than the productive causes that underlie the physico-chemical processes underneath at the lower level. So this gives us a much richer picture here of the genotype-phenotype map, actually not just with dynamic regimes, but with multiple levels in between. So basically, we have a picture now with constraints, default behaviors, feedback-driven processes, network dynamics, and higher level mechanisms. And they they amount to a much richer causal framework than the reductionist approach, narrowly focused on difference-making genes alone, okay? And for complex traits in general, this is the point of this lecture. It was exactly to this one conclusion. For complex traits in general, explanations at the level of regulatory processes are more stable, proportional, and specific than explanations based on the action of individual genes. So explaining developmental processes by the action of single genes is the wrong thing to do, because these explanations are unstable, sensitive to context and time. They are disproportional, so they're not relevant often at that level, and genes don't have specific effects because of the sensitivity of their effects, okay? So to put it another way, that's the same point. Although genetic explanations are more specific, that's true at the level of gene regulation. Remember, I said I did agree with Ken Waters' analysis of classical genetics, okay? The genes are the relevant factors there, but no longer at this level of complex traits where networks or interacting genes are more specific. Regulatory processes in general when it comes to complex traits. So the relevant level of explanation is not to bottom out and try to explain the behavior of the entire system at the level of single genes or factors, or even their sort of enhancers, their molecular structure. That's just missing the point, okay? If you're interested in how the network works as a whole, you need to look at the higher levels. You cannot ignore them, and you need to recompose the dynamical behavior of the system with a model. Otherwise, you cannot understand it unless the system is very simple, which is often the case just in those examples from classical genetics. Okay, so now we are going to move on in the next module to the ultimate challenge in terms of explaining a complex system. And that is, how can we explain the organization of an entire living being, its life cycle, and how it maintains itself, how it grows, how it reproduces, all of that. And it's mind-boggling, okay? So we've barely touched sort of the edges of this problem of organization of living systems. The big challenge is to explain the organism itself. So we'll go into that challenge with all the tools that we've developed, and with the spirit of this processual perspective that I've been developing all over the 7 first modules of this course. So I hope you will tune in again when we talk about organisms next time, and I hope you enjoyed this module. Thank you very much for listening. Goodbye now.